Okay, good evening, everybody. We'll be starting soon. Welcome to the weekly live interactive sessions of the course Genome Editing and Engineering, NOC24-BT71, which is taken by Professor Utpal Bora of IIT Guwahati. I'm Sanket Mukherjee, Prime Minister's Research Fellow from the Kusuma School of Biological Sciences, IIT Delhi. So today's session is mostly about zinc finger nucleases, and we are now getting into the genome editing tools that are used for gene editing and stuff like that. So I hope I'm audible and the annotations are visible. If it is so, you can just maybe react, or somebody can say if they want, if there's everything is visible and I'm audible. Yeah, fine. I'll just wait for a couple more minutes. Let others also join in, and then we'll start. Okay, let's start. So we'll to this a brief summary of what we have coming in this week is the basics of central dogma. We'll talk about central dogma of molecular biology. Then we'll talk about the protein structure and DNA binding fork-1 nucleases. So fork-1, which comes from Flavobacterium okeanocoides, this is a nuclease which we can use for cleaving any particular sequence. And then we'll talk about zinc finger domains. These are specific proteins which can recognize, which can bind to DNA. We'll see how we can add these two up, and we can I mean create uh, uh, an an artificial nuclease. So anyway, talking about the central dogma of molecular biology, it's very simple. Most of you must have studied about it. It's basically how DNA forms DNA through replication, right? I hope you all know. Uh, so the enzyme used in this process is the DNA polymerase. There are various kinds of DNA polymerases with with different fidelity and everything else. But the point is DNA forms DNA through replication. This DNA forms an RNA using transcription. That process is called transcription. If it happens in the reverse order where an RNA forms a DNA, it's called reverse transcription. Very common in various families of viruses like retroviruses, which have an RNA genome, but they form DNA as well. Well, RNA is then translated into a protein. So RNA encodes, it has all the codons which have the necessary information for each individual amino acid, which forms a protein. So this process is translation and it happens in a ribosome. So I hope you know how all these things happen. This is basically the central dogma. Now, talking about the protein structure. So now, so we know that a DNA that has the entire code of the genome, it forms DNA through replication, then it forms RNA, and then it forms protein. Now, these proteins are made up of amino acids. These are amino acids which are, in a sequence, it forms a protein. Now, a protein structure can be primary where, there is a linear polypeptide sequence of amino acid. That is where when we call a structure of a protein to be the primary structure. Now, this primary structure based on the, if you know how an amino acid is, the common, I mean, an amino acid is basically this is the basic structure of any amino acid. So R group changes based on the 20 amino acid. The R will change, but essentially each and every amino acid it has an amine group, it has a carboxyl group. That is, pretty much same. And what is important that since it has an amine group, which is, so it's an N, and then there is a C. That's why you will see proteins or peptides to be written at from N to C orientation. So all these things are important. Now, why I told you this, based on this R group, whatever is present, based on that, these primary structure of protein, they can form some secondary structure like alpha helices, beta sheets. These are some secondary structures which are formed due to hydrogen bonding between amino acids. Right. Now, coming to the third form, now tertiary structure is the 3D overall structure of this secondary structure. So when secondary structures, they form, a 3D structure. The secondary structure is usually, uh, you could say it would be two-dimensional, but when, for example, if a helix is there with the beta sheet and then there is more helix, you know, and like this, if these structures, they are superimposed together, it forms a 3D conformation. That kind of structure is tertiary structure. Now, these tertiary structures, they form a unit, and if these multi-unit complexes of structures, this is called a quaternary structure. So this is how um, different protein structures are there, how proteins are organized in a cell. So talking about alpha helix, it's the most common secondary structure. It's the most common secondary structure. And so if you see here, it's a helix basically. So essentially there are some things like there are 3.6 amino acid per turn, or what rather we could say that there is one residue at every 100 degrees. Right. And another thing is that every residue, if it is the ith residue, it forms a hydrogen bond with the I plus fourth residue. That is a characteristic of the alpha helix. So this hydrogen bond, how is it formed? So as we already know that proteins are formed of amino acid, which has the amine group and the carboxyl group. We know this. So the H in the amine, it forms the hydrogen bond with the O in the carboxyl group. So this is how hydrogen bonds are formed, right? So, well, so then there is beta sheet. So in case of beta sheet, it's a linear arrangement of proteins, right? So It's a linear arrangement of protein. And two adjacent beta sheets, if you see these, this is a beta pleated sheet, two adjacent beta sheet, they are joined by hydrogen bonds. If you see in the middle, they are joined by hydrogen bonds, right? And then these another important thing is that these sheets could be, in a parallel orientation or in an anti-parallel orientation. What does that mean? For example, N to C is the orientation in one of these sheets. The other sheet could be N to C again, or it could also be N to C like this. So if it is like parallel, so we call it as parallel beta sheets. It could be anti-parallel beta sheets also. Another thing which could be there is mixed. So there is not a particular orientation like there is parallel or anti-parallel. There could be mixed beta sheets as well. Well, okay. Um, So from there, why we are talking about proteins, because protein interaction drives cellular events, right? Even before before DNA was known to be the genetic material, the code of genome, protein was always considered to be one of the main factors which runs the cell, which runs an organism, because it has role in various you know processes. So if we talk about all the multiple processes which occur in our body, which are important processes, there are proteins which are involved. The role of DNA as such is not exactly seen. Although now we know that DNA codes for these proteins, so it is important for the DNA to be there as well. However, what is most important is that there are several DNA binding proteins, and these proteins are of specific interest to us. I mean, for this course specifically. Why so? Because these proteins will essentially, we can use these proteins to bind DNA, bind to specific DNA sequences. So that's if we know how these DNA binding proteins work. So in a review, it was already published that there are around 588 known DNA binding proteins, right? And there are out of these 64, they bind to both DNA. These are 64 proteins that bind to DNA and RNA both, right? So, and then there are 1210, like 1210 RNA binding proteins. So clearly, RNA is a little bit less stable than DNA. It requires protein anyway in its life in a cell. So what is the important, like what are the important proteins which are DNA binding proteins? So the proteins that are involved in transcription, packaging of DNA, like the histone protein, for example, DNA rearrangement, DNA replication, like the polymerases. The polymerases are a group of proteins which need to bind to DNA. And even the repair proteins that we have already discussed, there are several proteins which are involved in DNA repair pathways. Those proteins also need to bind to DNA for their function, right? So so 240, protein DNA complexes were studied. They were studied. And based on the structural analysis of these 240 protein DNA complexes, there were 8 major structural or functional groups of DNA binding proteins. So these are like helix turn helix. These are the groups. Zinc coordinating group, zipper type group, which is also called leucine zipper group. Then there is other alpha helix, beta sheet, beta hairpin or ribbon. There are others, and there are some enzymes also. which are there. So for example, if we look at helix turn helix, um, so it's basically like this. So helix, and then there is a turn, and then there is another helix. So these kind of proteins are involved in DNA um, binding. So multiple transcription factors, and there are multiple enzymes which have these domains, these helix turn helix or helix loop helix domains, which are important for DNA binding. So we will talk about an enzyme and restriction enzyme which comes from this particular species, Flavobacterium okeanocoides, and this restriction enzyme is FOC1. Fine. So it has, if you remember about the different classes of restriction enzymes, different types. So there was type 1, type 2, and type 3. If you remember, we had already discussed that type 2P is the most common restriction enzyme which is used in molecular biology laboratories. But this particular restriction enzyme is of type 2S, where S stands for shifted. P stands for palindromic, S stands for shifted. And what is the specific sequence of FOC1? It recognizes GGATC in the 5' end, and it cleaves 9 to 13 base pairs, 9 to 13 base pairs away from its recognition site. This is something which is known about FOC1 restriction enzyme. And it is again isolated from Flavobacterium okeanocoides. It's again a gram negative, non-motile. So so motility is actually not known. It could be motile, it could be non-motile, or motile, rod-shaped bacteria. Okay, from where we get this enzyme. And then, as we already discussed, it's a type 2S restriction enzyme. It has two distinct domains. One is the recognition domain. Fine. It has the N-terminal domain. As you know, any protein, it has the N-terminal and the C-terminal based on the amino acid structure. We already know that. So the N-terminal domain is the recognition domain. So N-terminal recognition domain. And then there is the C-terminal cleavage domain. How does this cleavage happens? This cleavage happens when both the domains, they form a dimer, and they basically, sorry, this is wrong. So cleavage happens when the enzyme binds to the recognition site, and in the presence of magnesium ions, there is cleavage of proteins. Now, the molecular mass of FOC1 is 65.4 kilodaltons. It contains 587 amino acids. FOC1 is type 2S restriction endonuclease. It consists of two functionally distinct domains. We already talked about this. And cleavage only happens in when there is dimerization. Okay, so essentially what happens, this is how it looks like. So whenever there is a recognition domain, so cleavage will only happen in the DNA if there is dimers which are formed. So essentially the cleavage domain, if you look at the protein like this, so if this is the recognition domain and then there is a cleavage domain which piggybacks on the recognition domain. As a monomer, this enzyme is catalytically inactive. It cannot cleave. As a monomer, it cannot. However, whenever there is formation of a dimer, that is when there are two FOC1s which are coming together, and at that time, this piggyback nuclease, domain, it comes and it sits on the DNA. This also sits on the DNA, and that is where they cause cleavage, basically breaks. are caused in the DNA. So you can read about it. There are several papers which are already referred to in the lecture, which talks about in detail how this enzyme works. But what is important for us to remember is that there is a recognition domain, there is a cleavage domain of this protein. And if we can manipulate the recognition domain or the cleavage domain, either of them, we can create a restriction enzyme which can cleave any of the sequence of our choice. So that's the idea. Okay. So approximately now, talking about zinc finger nucleases, zinc finger proteins, these are very important proteins in the proteome of a human proteome, basically. So in 1940, the biological role of zinc was established. And when we talk about zinc, we are meaning about Zn2 plus. So in 1985, there was discovery of in 1985, there was the structural role of zinc in in transcription factor 3A. So it was established in 1985, right? And this was from a clawed frog, African frog called Xenopus laevis. You must have read about it earlier also. So from this frog, they isolated the transcription factor and they found that zinc is involved, right? Then what we are talking about zinc finger proteins. These zinc finger proteins are very important proteins which are important in DNA binding. Now, what happens, it's basically a 30 amino acid repeat residue. So 30 amino acids are repeated. Out of these 30 amino acids, 25 are in the loop with the zinc in the middle. And the five which are remaining, they are basically linkers. So how it looks like is something like this. So 5 out of them are linkers and the rest 25, they are in this loop. And they are around the zinc basically ion. That's why they are called zinc fingers. This is how they look like, and that they bind to DNA. Um, well, these loops, they have conserved cysteine and histidine residues. And all these informations are already available in the lecture slides. So again, there are different zinc finger proteins like the C2H2 type, gag knuckle. These are different folds of zinc finger proteins which are already known. You can study about them. When we are talking about usually when we mention zinc finger proteins, we mean C2H2 type, which is C and H stands for cysteine and histidine, based on the conserved residue in the loop. So this is the most common type of zinc finger motifs. Okay, zinc finger motifs, they were first identified in the African clawed frog Xenopus laevis. We know this in the laboratory of Aaron Klug. A typical zinc finger, C2H2 zinc finger, contains a repeat of 28 to 30 amino acid sequence, including 2 conserved cysteines and 2 conserved histidines. Other combinations of cysteine and histidine as the zinc gelating residues are also possible. It's not that there is only 2 residues of cysteine and histidine. There could be more, but 2 are usually conserved. The regulation of transcription seems to be an important task performed by the C2H2 zinc fingers. So most of the transcription factors, they have these C2H2 zinc fingers, and they uh, mediate this DNA and protein interaction. However, recent studies have also suggested some protein-protein interaction of these proteins. Okay. Our C2H2 domain, it has 28 to 30 amino acids, which has a beta hairpin, and then there is an alpha helix. right? This is important, and they together form a beta beta alpha structure. So this zinc finger, this structure, this looks like a finger. These structures are stabilized through the zinc, essentially the metal ion. Due to the presence of zinc, these structures are stable. Uh, well, zinc, it plays a structural role and does not directly interact with the DNA. The alpha helix serves as a recognition element, inserting the DNA to major groove, where the side chains perform specific contact with three consecutive base pair. The overall interaction is further stabilized by non-specific contacts between beta sheet and sugar phosphate backbone. If we talk about the zinc finger proteins, just give me a moment. Yeah. There are zinc finger modules and then binding sites. A canonical zinc finger module, it consists of two anti-parallel beta strands, and then there is a alpha helix. With a zinc ion, which is coordinated with cysteine and histidine residues. Various amino acid residues at position relative to the amino terminal and alpha helix form important base contacts with DNA. We will talk about it in detail, how they form base contacts. Now, zinc finger proteins, they are DNA binding proteins, which has brief non-specific binding followed by semi-specific binding, and then there is specific binding. How is specificity ensured? If you look at the DNA like this, a normal zinc finger protein, how it binds to DNA. So if it is at this orientation from 5' to 3', the zinc fingers will be which will have main three domains. It will be arranged like this. So ZF1 domain, ZF2 domain, and ZF3 domain. Okay. Out of these zinc finger domains, the amino acids which are present at position 6, +3, and -1. These are the positions which are um, of the zinc finger domain. These amino acids are responsible for specificity of binding. So based on these three amino acid, it will bind to three nucleotides, for example. So based on the whichever amino acid is present in these three domains. So is what this gives us an idea that we can basically what we can do is we can manipulate these three amino acids, we can change them, and we can cause formation of different zinc finger proteins which can recognize different DNA. Now, this particular approach is called the modular approach, where we change the amino acid sequence and that changes the specificity. Fine. So there are publicly available, archives which has different plasmids which recognize different kinds of sequences. In 1996, a group of three scientists, Mr. Kim, Mr. Cha, and Chandrasekharan. These three scientists, what they did, they took zinc fingers, and they know that they can recognize any sequence of DNA, their sequence of interest, and they can, bind to it. The zinc fingers can bind to it. They used a zinc finger and they fused it with the FOC1 cleavage domain. So what happened now? After this, there was a chimeric protein because it comes from two different sources. That's why it's a chimeric protein, which can recognize a sequence of interest based on what you want, and it can cleave. So this is how there was creation of artificial nucleases. So again, there are different zinc finger nuclease and engineering platforms. So as we already discussed, it could be like the modular assembly method, right? Modular assembly could be one of the methods. Then there is CODA, context dependent assembly. Then there is, open method. We'll talk about these methods. And then there is some proprietary 2+2 method also, which is not known very well. So as we already said, in modular method, there are individual zinc finger motifs which are joined together so that you will get some it will basically bind to your sequence of interest. Okay. In case of context dependent method, CODA, basically in case of um, context dependent assembly, what happens is we know that some zinc finger, which because all these zinc fingers are basically all these sequences, they are already archived in the library. So when we know that, what is the efficiency when, suppose let's say ZF zinc finger 31, it works best when it is paired with zinc finger 28, like this. These kind of information is known. So it is a context dependent assembly. So that is CODA. And again, then there is a high, specificity or high activity of these zinc fingers. Then there is oligomerized pool where there is a open source combinatorial selection of zinc fingers. Now, coming to, we know we can design a zinc finger. Now, how to deliver these into the cell? So delivery is also very important. So I don't know if you can see this. Okay, I see the slides are stuck. And try to. Don't know what's the issue. Yeah, now it's working. Okay, talking about the delivery strategy. So we know that about zinc finger nucleases. These are nucleases which can work upon a specific DNA sequence and it can cleave them. So that's what we know. But how to deliver these zinc finger nucleases into the cell? So there are various methods. One is like the viral vectors. So if we talk about viral vectors, we are talking about different viruses like adenoviruses, or we are talking about adeno-associated viruses, or lentiviruses, which are routinely used for normal insertion of genetic elements. These kind of vectors can be used for delivering the zinc finger motifs as well. There are non-viral vectors also. If we talk about non-viral vectors, they are like polyethylene amine. Polyethylene amine is a transfection reagent. Polyl lysine. Then there are some cell-penetrating peptides which can get inside the cell, and it can also take your protein along with you, or the protein coding gene along with you. Apart from that, there are some physical methods also. If we talk about the physical methods, they are like electroporation, microinjections, all these kind of things. These are used to deliver the required protein or DNA of interest. So how is the design strategy? Basically, if we are talking about, let's say, what was done by the group of Chandrasekharan, where they took a FOC1 cleavage domain and they cleaved it with basically the zinc finger recognition domain. How to do that? We can do that by creating fusion proteins. So how we do, if we take the mRNA of these two genes, if we take the mRNA and we fuse them. So if we fuse them together, so this is the first mRNA and this is the second mRNA. We fuse them and we take this into a cell where it is translated using ribosome. Ribosome translates basically mRNA into protein. So it will give you a fusion protein which will have both the proteins, protein A or one and protein B, both together. That kind of thing can happen. We can do. I mean, that is the design strategies for creating chimeric, restriction enzymes or nucleases. Talking about the applications, what are the, applications? So basically, any kind of gene experiment is done to understand the biological function of a gene and to look at some novel phenotypes which are usually rare, cannot be seen usually. We can create those kind of phenotypes through gene editing. Now, what are like the applications are like creating mutations by gene insertion, deletion, all these kind of things. We can do large deletions of genes. We can do that. There could be application like crop improvement. Like crop improvement can be done through um, modifying the gene using a zinc finger nuclease. We can perform gene replacement, obviously, using zinc finger nucleases. Then there is HIV treatment. So if you already know that in case of HIV, there are two major receptors, CCR5 and CD4 plus. These two main receptors are involved by the T cells. They internalize the hepatitis. So the HIV virus. So they internalize it because of these two receptors. If these two receptors can be disrupted, then HIV internalization will be disrupted. So HIV treatment could also be one of the applications. Then in case of cancer, where there are multiple gene changes, there are mutations which happen. We can potentially change these things using zinc finger nucleases. But however, we will realize later that there are zinc finger nuclease. Using it is a very tedious process, and ideally, there are multiple other advancements in the field, like there are talons, and then there is the CRISPR, the system of CRISPR, which gives you much more freedom in gene editing. We'll discuss about those things in the upcoming lectures. So let's talk about the interactive part of this session. So I would expect some of you, to either answer this either in the Q&A part, or you can just speak if you know the answer. That's also fine. We already have less students today. So question one, which is the most common type of secondary structural element in proteins? Is it alpha helix? Is it beta sheet? Is it parallel beta sheet or anti-parallel beta sheet? Anyone would like to answer? No answers? I don't think the people who usually answer, they have joined today. Anybody? Nobody if is answering, then it's alpha helix. We already discussed alpha helix is the most common type of secondary structure. Zinc fingers were first identified in a study of transcription in the species of an African frog, African clawed frog, an African threadfish, or an African wild dog.
Option B.
Yes, you're right. Option B. The proposed sequential binding scheme of 3-cysteine histidine zinc finger domain bound to the cognate DNA are specific binding, semi-specific binding, non-specific binding, or non-specific binding, specific and semi-specific, or non-specific, semi-specific, and then specific, or binding, specific binding, semi-specific binding.
Non-specific, semi-specific, and specific.
Right, you're right. Which is not the current technique used for gene delivery in zinc finger nuclease. Viral vectors like adeno-associated virus vectors, adenovirus vectors, and lentiviral vectors, non-viral vectors containing polymers and cell-penetrating peptides, physical methods containing microinjections and electroporation, or GFP or molecular scissors.
Option D.
D. Yes. Yes, you all are right. So we have already discussed about it. It's already there in the slides also. These are the techniques used for gene delivery. We have already discussed about it. So this is an excerpt taken from your slide. So where we could deliver through viral vectors like adenoviral vectors, adeno-associated viral vectors or lentiviral vectors. Then there is non-viral vectors like polyethylenamine, polyl-lysine, liposomes, and all those kind of things. And there are physical methods also. We have already discussed this. The CCR5 delta 32 mutation was initially discovered in 1996 as a genetic mutation that that confers protection to cells from infection of coronavirus, HPV or human papilloma virus, HIV, immunodeficiency virus, human immunodeficiency virus, or smallpox. All of these are viruses.
HIV.
HIV, yes, as we already discussed, if it's again there in the slide, you can already study about it. So there are different glycoproteins which are present in the cell, and these in the virus basically, which bind to the CD4 and CCR5 receptors. When these receptors are mutated, internalization of HIV does not happen. The virus cannot replicate, cannot in the cell. So this mutation was initially discovered, and these kind of when there was a deletion in the CCR5 gene, these patients were rather immune to HIV infection. And then there were several studies. Even there was a very controversial study. Very recently, it came out from China, where they tried to, create this mutation in a newborn baby, in a human, in a girl. And they claimed that this girl is HIV resistant. She won't have HIV. But there were multiple ethical concerns that came up because of this, because how would you even verify that any person who has this mutation, or if you have created this mutation and created a human being out of it, how would you even verify that they are really resistant or not? You have to infect them with HIV, and that in itself is not ethical. So anyway, that's not the idea. But the thing is, these two receptors are involved in internalization of the virus, and then they replicate in the cell. Which of the following is famously known as the guardian of the genome? Is it P63, D53, E6, or P53?
P53.
Yes, P53 also, it's also a very important tumor suppressor gene. It's called the guardian of the genome. So it's a 53 kilodalton protein. So zinc finger nucleases were first used against DASH, which prevented the virus from replicating within the host cell. HPV 1618, HPV E2 gene, HPV 16E6 gene, or HPV P63.
Option C.
So they were used first against HPV E2 gene, which prevented the virus from replicating. So there are various gene elements in the open reading frame of the virus. When E2 was deleted from this virus, the replication was compromised. That is what it was seen. And HPV is basically human papilloma virus. I hope you know about it. Which of the following is incorrect in zinc finger nuclease mediated targeted genome modification via double-stranded break repair? A pair of zinc finger nucleases bind to a target sequence, dimerize through FOC1 domains, and join the DNA at a defined site. The DSB generated by zinc finger nuclease cleavage induces the DNA repair process. In the absence of donor DNA, error-prone non-homologous end joining happens. Or D, in the presence of homologous sequence, homology-directed repair happens. Which of the following is incorrect?
A.
Sorry.
A. Option A.
So yes, let me. I have to see this. So this is the zinc finger nuclease mediated targeted genome repair. So a pair of zinc finger nucleases that bind to the sequence dimerize through fork1 domain and cleave the DNA at a defined site. Then this DSB is generated by a zinc finger cleavage that induces DNA repair process. In the absence of donor template, there is non-homologous end joining and targeted mutagenesis. In the presence of homologous sequence, there is homology-directed repair, which results in gene editing or site-specific integration. So what was wrong here? A is wrong. You're right. Classical zinc ribbon belongs to which fold group? C2H2 type, um, gag knuckle, treble clef, or zinc ribbon? Any answers? It belongs to the zinc ribbon group only. Zinc fingers were first identified in a study of transcription in the African clawed frog. What was the species? The name, scientific name. Was it A, Xenopus laevis, Xenopus tropicalis? Uh, this is Pixicephalus adspersus or Amytophrynus regularis.
Xenopus laevis.
Right, you're right. How many repeated amino acid sequences does a typical C2H2 zinc finger contain? Is it 38 to 40, 28 to 30, 28 to 30, or 15 to 20?
28 to 30.
Actually, it should be 28 to 30. So as we are, there are actually 30 usually. It could be 28 as well. So out of which we have already studied that 25 of them are involved in the loop and 5 are linkers. So anyway, which of the following is the rate limiting step in homology directed repair or homologous recombination for gene modification? Is it gene targeting, generation of non-specific desired genomic double-stranded breaks, generation of specific desired genome double-stranded breaks, or none of the above? This is the question. Any answers? Generation of specific desired double-stranded breaks. This is the main rate limiting step of this entire homologous recombination. The current techniques used for gene delivery, is it viral vectors like adeno-associated vectors, adenovirus vectors, and lentiviral vectors, non-viral vectors, or physical methods, or all of the above? Which are the current techniques used for gene delivery? Any answers?
Option D, all of the above.
You're right. Thank you for participating. Now, what is ribosomal skipping? Mechanism of translation in which specific viral peptide prevents the ribosome from covalently linking a newly inserted amino acid and let it continue translation, resulting in co-translational cleavage of the polyprotein. Mechanism of translation in which a specific viral peptide promotes the ribosome to covalently link to a new inserted amino acid and prevent translational cleavage of the polypeptide polyprotein. Mechanism of translation in which an indigenous non-viral peptide prevents the ribosome from covalently linking a new inserted amino acid, resulting in co-translational cleavage of the polyprotein. Or D, mechanism of translation in which a non-specific peptide prevents the ribosome from covalently linking a new inserted amino acid and let it continue translation, resulting in co-translation cleavage of the polyprotein, which would be the answer. Ribosomal skipping.
B.
A would be the answer. Mechanism of translation in which a specific viral peptide prevents the ribosome from covalently linking a new inserted amino acid and let it continue translation, resulting in co-translation cleavage of the polyprotein. So what essentially happens if you know how ribosomes work, so this is the mRNA, which has the codons like A, T, G, and all those kind of things, which are used, which are recognized by the anticodons, which are present in these ribosomes, right? These anticodon groups, they look like this. We know this anyway. So it's present, and this ribosome, they link the newly formed amino acids. They are linked together through covalent bonds. Now, this is how this entire translation process works. But when there is a viral peptide, there is some other peptide, it prevents this covalent bonding to happen. So what happens if this is, let's say, the gene? So this is the gene, and it codes for an mRNA, and this will code for some protein, so which will have a sequence of amino acid. Okay. But what happens because of this disruption in covalent linkage, there is a co-translational cleavage. So what happens? Let's say till here, um, the RNA is forming a protein, but this part is cleaved off. This part is not forming any protein because of that absence of covalent linkage, which happens due to the presence of a viral peptide. So this kind of cleavage is called co-translational cleavage. So is it clear to everybody? And this process is called ribosomal skipping. Is it clear?
Yes, sir.
Okay. Which of the following statements about direct delivery of zinc finger nuclease proteins is true? Zinc finger nuclease can penetrate cell membranes due to positive charge of cysteine, histidine, zinc finger domains. Zinc finger nuclease can penetrate cell membranes due to the negative charge of the cysteine, histidine, zinc finger domains. Zinc finger nuclease can penetrate cell membranes due to the absence of any charge in the C2H2 domain, or zinc finger nuclease cannot penetrate cell membrane due to the absence of charge. What would be the right answer?
Has positive charge.
You are right. You are right. Which of the following statement about gene deletion using zinc finger nucleases is true? Gene deletion can be achieved by a single zinc finger nuclease mediated double-stranded break. Gene deletion cannot be achieved using zinc finger nucleases. Two concurrent zinc finger nuclease mediated double-stranded break can result in the loss of a gene, or none of the above.
C.
You're right. With which of the following strategy a pair of zinc finger nuclease can be cloned and co-expressed together in a single vector? Increase the length of polyadenylation signal, introduction of a two-way sequence, introduction of long terminal repeats, or inactivation of promoters.
Introduction of 2A.
You're right. In the presence of homologous sequence, homology directed repair or homologous recombination results in dash or dash. Is it gene editing or site-specific integration? Targeted mutagenesis or site-specific integration or gene editing, which is basically the first option only, and gene editing and targeted mutagenesis. It would be option A or option C, anyway, how you would like to see. So that's all we have for today's session. Do you have any questions from this week? You can ask now if you have any questions. I'll stop the recording at this point.